Okay, let's get started. Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to review. Questions will be responded to at the end of the presentation. Please enter your questions in the Zoom Q&A box. The audience can prioritize the order of the questions by clicking on the thumbs up button. If you could, closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions will also be provided in the chat box. I should also note these sessions are being recorded. The recordings are usually up about 2 weeks after today's presentation. So if you want to refer back to the recordings for anything that you might have missed. With that, I would like to turn the proceedings over to our moderator, Dr. Derek Abbott. Dr. Abbott, graduate with a bachelor's degree from the University of Virginia before embarking on an MD and a PhD training at Vanderbilt University, where he studied breast cancer genetics. After finishing medical school, he undertook clinical training at the Anatomic Pathology at Brigham and Women's Hospital, and then a postdoctoral fellowship with Lewis Cantley at Harvard Medical School. In 2006, he moved to Case Western Reserve University to start his independent lab. Dr. Abbott's lab studies the interface of genetics and immunology with an emphasis on autoinflammatory disorders and immuno-oncology. He is a recipient of a Burroughs Wellcome Career Award from the Burroughs Wellcome Foundation and the Adolph Adams Award from the Society of Leukocyte Biology. He was a director of Case Western's medical scientist training program for 7 years, serving as the primary director for 3 years. Currently, he is the Curtis F. and Arlene H. Professor of Medicine at Case Western Reserve University. Dr. Abbott.
Thanks, Alan. So I'm privileged to introduce Keith Sison-Chan for this seminar. Dr. Chan received his PhD training at UT MD Anderson Cancer Center and postdoctoral training in Irving Weissman's lab at the Stanford Stem Cell and Cancer Institute, where he investigated epithelial cancer stem cells and how they evade immunomodulation. In 2009, he started and positioned his independent lab to focus on translational cancer research. His lab is part of an international consortium in the subclassification of bladder cancer into clinically relevant subtypes. Additionally, his lab is taking a holistic view to dissect how the interactions between cancer, stromal, and immune cells within the tumor microenvironment contribute to therapy resistance using single-cell and spatial technologies. Most recently, his lab investigates the paradoxical roles of cell death-induced biology in modulating therapy resistance via promoting compensatory proliferation and immunosuppression. He is the recipient of an NCI K99R00 Howard Temin Award, a V Foundation Scholar Award, and a CIPRIT Established Investigator Award. Currently, he is a professor and translational research director at Houston Methodist Urology and the program lead of innovative therapeutics at Methodist Neal Cancer Center. He's excited to team up with clinical colleagues to expedite research into investigator-initiated trials to benefit cancer patients. Dr. Chan?
Thank you, Derek, for the introduction, and thank you for joining this webinar. And it's an honor to be presenting our work here. Can everyone see the screen? Okay.
Yes. Yes.
Okay.
Good.
Perfect. So, as Derek mentioned, when I started my independent lab, we really focus on performing translational research that hope to bridge basic science discovery into early phase clinical trials. With my relocation to Houston Methodist, I really hope to team up with vibrant colleagues where we can obtain tissue from the clinical trials back to the clinic to complete this circle. So for today, I would like to talk about some serendipitous findings that lead us down the road to some very exciting role of understanding the paradoxical role of cell death in modulating biology that can impact therapeutic response. So as a cancer biologist, similar to all of you here and in the audience, that for decades, we were trained to understand the mechanism and also design strategies to induce programmed cell death. So in a two-part talk today, I hope to convince you that there are tremendous biology that is induced by cell death. In part one, we talked about compensatory proliferation or repopulation of residual cancer stem cell, a phenomenon that is originally observed in Drosophila in the 70s in response to radiation. And in part two, we would like to introduce our data on cell death-induced immunomodulation or immunogenic cell death, a term that is coined by Guido Kromer, Doug Green, Stephen Tate, and other colleagues in the field. So to step back and introduce the cancer type that we are most focusing on is bladder cancer, which is a very understudied cancer type, but the second most common urological malignancy after prostate cancer. From a schematic taken by Maginon's beautiful review, showing that the clinical staging of bladder cancer is really measured by the invasion or depth of invasion into the lamina propria, the inner muscle layer and outer muscle layer into the perivesical tissues. In the field, the investigators has been subdivided bladder cancer into two major subtypes. The TAT1 classified as the non-muscle invasive bladder cancer, and the T2 and higher subtype has been shown regarded as the muscle invasive cancer. It's been shown that there are different pathogenesis and genetic alteration driving these two types of cancer. And as you would imagine, increasing T stage associated with a progressively poor outcome for the patient. For the time being, we will be focusing on the muscle invasive bladder cancer today. So muscle invasive bladder cancer before 2017, the standard of care remained neoadjuvant gemcitabine cisplatin chemotherapy, followed by radical cystectomy, which is the surgical removal of the entire bladder. Data here adapted from meta-analysis showing that 11 trials, over 3,000 patients, clearly demonstrating that neoadjuvant chemotherapy have a significant but modest effect in improving patient outcome. So therefore, the mechanism leading to this chemoresistance is a major clinical gap in the field. So in the field, investigators use various models for studying chemoresistance. A very commonly approach in the lab is to expose cell lines to long-term selection of chemotherapy, leading to this resistant clone. And by comparing the resistant clone versus the parental clone, I can understand a lot of molecular biology that leading to this chemotherapy resistance, or by subjecting xenograft to short-term chemotherapy treatment. With these strategies, investigators in the field found multiple classical mechanisms of resistance that are primarily tumor cell intrinsic. For instance, there are so-called pre-target, on-target, or post-target effect that cancer cells decrease the reporters that is expression of reporters that is important for uptake of the drugs, or they overexpress the so-called multiple resistant drug resistant transporter that efflux drugs, particularly in cancer stem cells. There are, for example, ABCG2 or other multiple drug resistant transporter that are overexpressed to efflux drug out. Also, they found it's been shown that there is increased DNA repair mechanism or increased tolerance to DNA lesions, leading to chemoresistance. And finally, but there's a lot of data showing that there's defect in the cell death or cell senescence pathway that drive a pro-survival signal for chemoresistance, such as overexpression of BCL-XL and BCL2 proteins. However, in the clinic, the patients are treated in multiple cycles of chemotherapy with gap period in between, allowing for the bone marrow stem cells to recover to minimize toxicities. So we rationalized at the time that if we follow these clinical regimen of treatment, we might find additional biology related to the clinical development of resistance. So my former student, Antonina Katofer, and my former fellow, Jing Xiao, obtained xenograft and generated xenograft derived from patients that do not respond to chemotherapy. And they subject these xenografts to clinical regimens of chemotherapy, multiple cycle of chemotherapy treatment. To our surprise, at that time, compared to vehicle control, the first cycle of chemotherapy seemed to show response. But it's not until the multiple cycle of chemotherapy where you see the manifestation of chemoresistant phenotype. And this phenomenon can be reproduced in additional patient-derived xenograft, as well as other immortalized xenograft models. So to cut the long story short, we found that chemotherapy, similar to a severe wound injury situation, where injury can induce a repopulation of primitive stem cells to repair the wound. Chemotherapy also, while they induce programmed cell death of the downstream differentiated cells, they secrete factors that can lead to repopulation of quiescent stem cells into repopulation. And with multiple cycles of chemotherapy treatment, they lead to accumulation of cancer stem cells that manifest the chemoresistant phenotype. So here is the raw data showing supporting our findings that after GC chemotherapy treatment over time, you can see a reduction of fibrous cells, which is associated with an upregulation of cleave caspase 3, indicative of apoptosis, as well as temporal upregulation of COX-2, the enzyme responsible for metabolizing arachidonic acid into PGH2 and into PGE2. And you can see there is a cell death associated increase of PGE2 release into the supernatant measured by ELISA following GC chemotherapy treatment in this bladder cancer cell line. And this phenomenon is a generalized phenomenon since we see this is a dose response. You can see increasing dose of GC lead to increase of release of PGE2 into the supernatant. And with multiple cell lines, we can see there's a clear association of PGE2 release into the supernatant associated with cell death. Very gratifying. Another group from UK led by San Diego, SLAB, also show that in another type of chemotherapy agent, and in multiple different cancer cell types, such as colon, breast, and melanoma, that there's a cell death associated induced release of PGE2 into the supernatant, suggesting that this is a generalized phenomenon that is not only limited to our model. To study the effect of this PGE2 release into the supernatant or into the extracellular space, we designed first an in vitro assay to assay sphere-forming stem cells indicative of cancer stem cell frequency. So we obtained the supernatant that are treated with chemotherapy, and which encapsulate the factors that are being released by chemotherapy-induced cell death. We put them into this sphere-forming assays in the presence or absence of neutralizing antibody or the FDA-approved drug against celecoxib, against COX-2 celecoxib. And the data here you showed has shown that the supernatant containing these factors that are released significantly induce fear-forming stem cells, which can be diminished by both the neutralizing antibody against PGE2 as well as celecoxib with the IgGS control, indicative that the PGE2 is one major factor in driving cancer stem cell proliferation and expansion. To study this in vivo, we utilize a classical approach to label stem cells using label retaining cells and a double labeling with another analog. Following chemotherapy treatment, we can follow the effect of chemotherapy in driving the division of slow cycling stem cell as well as the newly incorporated proliferating cells. So here at time 0, you can see that the slow cycling cell is mutually exclusive with these proliferating cell as expected. In 6 hours following chemotherapy treatment, you can see there's a significant drop in proliferating cells which is expected since the gemcitabine is expected to incorporate into proliferating cells is an analog that target proliferating cells. But interestingly, at 12 hours after chemotherapy treatment, you can see that there are cells that are seems to be dividing, incorporating both the proliferating cytidine, which shows that cells that are dividing. And you can see this repopulation of slow cycle proliferating cells that coincide with the expansion of cells that are double label, indicative of quiescent cells being proliferating and coincide with the expansion of cancer stem cells marker. So these data really demonstrate that in vitro and both in vivo that the stem cells are being recruited into proliferation and likely by factors that are being secreted by the dying cells. So we further utilize a FDA-approved drug celecoxib that target is COX-2. In combination treatment, we found that the combination treatment of COX-2 with GC really sustained the response in this resistant PDX. And when we look at the content of this xenograft, we really see that in the combination treatment, the GC-induced expansion of cancer stem cell is significantly reduced compared to the GC treatment alone. In another model, T24 model, where they form metastasis, we can see similar effect in the primary tumor, as well as in the lung metastatic nodule. You can see that combination whilst GC chemotherapy is effective in reducing the overall metastatic lung burden, combination therapy is significant in further reducing this metastatic burden shown here. So these data really hope to convince you that chemotherapy-induced release of PGE2 play a role in recruiting the quiescent stem cells into proliferation. And FDA-approved drug can block these effects and contain the expansion of cancer stem cells, as well as sustain the response to chemotherapy treatment. So we have now extend our study into single cell analysis in other models and try to look at the granularity of different population of cancer stems, progenitor, and differentiated cells, and hopefully to understand this phenomenon further. Interestingly, at the time, there's also another study in the canine published by Deborah's group in Purdue showing that a non-selective celecoxib cyclooxygenase inhibitor really reduced the enhanced the response of vinblastine chemotherapy by more than 30%, really encouraging results. And we have started our trial with single arm trial combination with the celecoxib to try to see this phenomenon in vivo. However, we know that the limitation of our previous preclinical study, because it was the PDX, while it was at the time one of the most relevant model, clinical relevant model for studying chemoresistance, they were performed in immunocompromised setting without taking into the account of the immune microenvironment. So this moved into the part 2 of the talk, that we are now interested in looking at the immunomodulatory role due to the recent emerging success of the immune checkpoint blockade therapy. So in 2017, bladder cancer received expedited approval of the immune checkpoint blockade therapy. And this therapy revolutionized a lot of tumor types, including melanoma, first in melanoma, lung cancer, as well as bladder cancer. And the response rate is quite remarkable, with a 21% to 30%, depending on the trial. So like to step back and introduce how these immune checkpoint blockade therapy works. So to achieve T cell activation, the APC or tumor cell has to express MHC that binds to T cell receptor as so-called signal 1. And there is CD80 or 86 that bind to CD28 as a co-stimulatory activation as a signal 2. In cancer, or with chronic antigen exposure, the cancer cells will overexpress the PD-L1. And in bladder cancer, Turley's group, as well as Tom Powell's group, shows that the myeloid cell or the stromal cells overexpress this PD-L1 that binds to PD-1 on T cells that blocks signal 1. And activated T cell, shown by the Nobel laureate Jim Allison, that they will express CTLA-4, which competes away these CD28 co-stimulator molecule to block signal 2. So this class of immune checkpoint blockade therapy, namely anti-PD-L1, PD-L1, as well as anti-CTLA-4, really have the goal to remove this co-inhibitory signal and to unleash the T cells so that they can target the patient's own T cells to target the tumor. So to improve this patient response, multiple clinical trials, large-scale phase 3 trials, aim to combine standard chemotherapy with immune checkpoint blockade therapy. Unfortunately, both the IMvigor 130 and Keynote 361, two very highly anticipated trials, essentially result in very undesirable outcome. So this is one of the trial led by Matt Gelsky's group, showing that the objective response of the combination chemotherapy and ICI essentially is not very significantly benefit over single chemotherapy treatment. In fact, the prolonged response of ICI is even nullified by the combination treatment. So while at the time it was the rationale was to use chemotherapy to release new antigen that hopefully will have additional effect of this combination therapy. In retrospect, it seems to be counterintuitive because cytotoxic chemotherapy are also known to have severe lymphodepleting as well as myeloid depleting effect. So our lab dig into the basic biology a little bit more. So here is a review from Lorenzo's group showing that, in fact, not all chemotherapies are inducing this immunosuppressive effect. A handful of agents such as oxaliplatin or cyclophosphamide are known to induce so-called immunogenic cell death and able to combine with other therapy to enhance T-cell response. However, the standard of care chemotherapy, cisplatin and gemcitabine for bladder cancer are not among these immunogenic cell death reagents. So we have performed a mass spec proteomics profiling the supernatant of chemotherapy-treated cells. At the time, in retrospect, we were trying to identify additional mitogen that will induce cancer stem cells. But surprisingly, what we found is that in the supernatant, there's a class of a structurally unrelated protein, namely, for example, calreticulin, HMGB1, or these heat shock proteins that are known to be so-called damage-associated molecular pattern. So to step back and introduce this phenomenon, this is work done by my former student, Kazukuni. So the immunogenic cell death is a phenomenon known to be induced by the several molecular signatures, such as translocation of calreticulin from the ER to the cell surface as so-called eat me signal to recruit dendritic cells and other antigen-presenting cells. And there are also the release of, as I mentioned, the HMGB1 or other heat shock protein as DAMs or danger signal that engage the pattern recognition receptors on dendritic cells, rendering them to mature properly and prime a CD8 positive T cell response. So the current dogma suggests that if there is DAM release to engage this DC and CD8 T cell priming, there will be immunogenic cell death. But to our surprise, in the supernatant that are treated with two agents, gemcitabine and cisplatin, we found these DAM release. We later confirmed by flow cytometry as well as by Western, confirming our mass spec data. So to address this initially puzzling phenomenon, so we turned to a classical vaccination assay that's to analyze immunogenic cell death reagent. So we this is how the assay works. So we will treat the cancer cells with IC50 dose of the drug. So if the drug is a ICD-inducing agent, and then we inject them as a vaccine into the left flank of the mice. 7 days afterwards, we will come back with the live cancer cells as a challenge. So if this is a ICD-inducing agent, it will mount a proper DC and T cell response, and there will be to reject the tumor, and there will be high percentage of tumor-free mouse. But if this is a non-ICD-inducing agent, then it won't be able to reject the challenge of live cancer cell, and tumor will form. So to evaluate the molecular inconsistency at the time, we look at how gemcitabine and cisplatin compared to a positive control. So as expected, the control mitochondrion rejected non-ICD inducing agent rejected the tumor formation, while the single treatment of gem or cis, they are non-ICD inducing agent, despite the fact that they are releasing these DAMs. So we thought that at the time, that DAMs may not be the only determinants for determining immunogenic versus tolerogenic cell death. And based on our previous experience, we know that gemcitabine also induced COX-2 and PGE2 release. And there was a paper using freeze-thaw profiling indicating PGE2 is a tolerogenic factor. So we hypothesize that ICD induction is not simply determined by DAMs in activating the pattern recognition receptor, but it's a balance of both of the DAMs and PGE2, which we call inhibitory DAM, to determine the tolerogenicity and the immunogenicity. So to address this, we again turn to celecoxib to remove this IDAM and see what's the effect in the functional assay for the vaccination assay. As you can see here, combination treatment with drug or using a genetic depletion of COX-2 really convert gemcitabine into a non-ICD agent, inducing agent into a agent that can reject tumor formation. And using a CD8 positive antibody depletion, we confirmed that this phenomenon is CD8 positive T cell dependent. So to address the mechanism further, so we adapted a footpad injection assay with Sokazu in collaboration with Fotis in the lab, adapted this vaccination assay. Now when we inject drug-treated dying cells into the footpad, so we can look at the lymph node, the draining lymph node, the status of dendritic cell frequency as well as activation. So with this gating strategy, we can see that the effect of celecoxib or inhibiting this pathway really induced the frequency of CD103 positive dendritic cells draining into the lymph node. More importantly, it induced signal 1, shown here by MHC class 1 as well as MHC class 2, signal 2, CD40, CD86 significantly induction, as well as signal 3, the cytokine release of IL-12, as well as the lymph node trafficking signal CCR7. So these data really shows that IDAM blockade really favor CD103 positive dendritic cell activation and draining to the lymph node. So I hope that I have convinced you that by removing the IDAM leading to the maturation of dendritic cells. And what about the T cell response? So at a different time point, Kazu and Fotis also looked at the effect of TC1 response indicated here and clearly show that there's a dramatic TC1 response and a diminished Treg response, while the activity of T cells are significantly enhanced in this setting. So then with these data in mind, we would like to move into a preclinical setting where we can reproduce a clinical regimen of chemotherapy treatment. So just have some background here showing you that in the field, it's been shown that there are at least several types of immune microenvironment, the so-called inflamed T cell inflamed tumor that is associated with favorable response to ICI treatment. And there are other immune excluded or immune detected tumor that are less responsive to ICI treatment. However, in the field, majority of the tumors that are being used for preclinical investigation inflamed tumors. So our lab have developed our own model called the G69 bladder cancer cells that is representative of an immune excluded tumor. You can see that the T cell marked by green color here. See the T cells are being excluded in the periphery or within the stromal region that are marked by the fibroblastic marker here. So we rationalize that this is a good model that recapitulate patient response. In fact, when we subject this xenograft into GC chemotherapy, we found that this is a resistant model, and it's also highly resistant to PD-1 or PD combination chemoimmunotherapy. And intriguingly, when we add the celecoxib to block the IDAM release, there's a significant effect. And when we combine that with chemoimmunotherapy, there's a further reduction of tumor volume and a sustained response. And we don't have time to show all the DC and T cell data, but here just to show that there's activation of T cells with the combination treatment. And interestingly, when we look at spatially, you can see that when the tumors are restricted outside in the GC treatment, GC plus the IDAM blockade really lead to infiltration of T cells into the tumor core, flipping a immune excluded tumor into a more inflamed tumor. And these are the graph quantifying the T cell density normalizing from the distant from the periphery. And you can see that GC with the IDAM blockade really move the T cells into the center of the tumor. And functionally, when we deplete these tumors with the CD8 positive T cells, you can see that the T cells are shown to be functional, while in the compared to the non-depleted control. So this is a more global view showing that the combination treatment and chemoimmuno combination with the IDAM blockade, showing that there's a tremendous infiltration of T cells that are activated into the tumor region to exert their functions. And most importantly, this pathway is clinically relevant because we mine the data, the pre-treatment tissue data from Invicta 210, which is treated with a immune checkpoint blockade therapy. And we find that a high PTGS2 or COX-2 level in the pre-treatment tissue of patient associate with a poor response. And patients with a high expression of PTGS2 also correlate with a poor survival. So very gratifying that these findings are being reproduced by other group, that in the breast cancer model, that combination treatment with celecoxib really enhance the chemoimmunotherapy response, both in the primary site and in the metastatic site. So I hope I have convinced you that in addition to the dogma that we think that postulate that DAMs are the only determinants for determining an immunogenic response that lead to immunogenic DC maturation to primer T cells, we found that it's a balance between these DAMs and inhibitory DAMs, and that to determine immunogenic versus tolerogenic DC maturation. And with this strategy to remove IDAM, we can really sensitize tumor response in the chemoimmunotherapy treatment setting. And now we have Stephen and Ethan in the lab is really trying to see if we can generalize this DAM and IDAM concept into other drugs that are being used in the clinic. For example, there's a investigator in NCI using Andrea Polo using the cabozantinib to combine with ICI, showing that initial response. We found that this drug also induced high level of COX-2, suggesting that it might be a strategy to combine combination of COX-2 inhibition with this drug to further enhance the immunologic response. And another more immunogenic agent, radiation and the mitoxantrone, you can see there's a significantly lower COX-2 induction. So we are in the process of performing the functional assay to drive this point home. And very excitingly, our clinical colleague, Dr. Raj Sakunas Visan in the Methodist is really driving a trial with a radiation combination ICI, and with some initial success. I very much look forward to obtaining patient tissue from his trial to see whether our concept is also validated in patient tissues. And obviously, we have our own interest to try to combine this drug, drugging this pathway in combination with chemoimmunotherapy. So we are collaborating with Tempus, where there's a drug that blocks the PGE2 receptor 2 and 4. And we are really seeking drug sponsor for ICI in order to perform this combination trial. So if you're in the audience, you have industrial sponsor, really look forward to collaborating with you. So I hope to summarize the talk today to convince you that there is cancer cell death is not necessarily the end game. So in the first part of the talk, we showed that there's cell death release of PGE2 that act as a mitogen to induce cancer stem cell repopulation. And there's also release of DAMs that prime a DC, that's important for priming a DC CD8 positive immunogenic cell death. And PGE2 acts as an inhibitory DAM to nullify this function as non-classical mechanism of resistance. And we are on our way to move forward to clinical, multiple settings of clinical studies to evaluate the DAM-IDAM concept and whether removal of this pathway can enhance chemoimmunotherapy response. And just to show some preliminary work from Beth, that this phenomenon not only end in the epithelial compartment or the immune compartment. We have data suggesting that it can this cell death, chemotherapy-induced cell death, also played a role in modulating the extracellular matrix microenvironment that might then feed back to leading to the immune exclusion and other phenotype. This is very exciting because it circled back to our other data in the lab, really looking at the stromal, ECM, and collagen microenvironment in the primary tumor microenvironment, as well as in the metastatic microenvironment. And just want to show the last data slide here, showing you that obviously in this era, programmed cell death is not only limited to apoptosis. There are other type of cell death, such as the pyroptotic, caspase-1 dependent pyroptotic cell death that can be mediated by cleavage of gasdermin D that form pores, and another type of cell death known as necroptosis. There's regulated by multiple upstream, leading to the phosphorylation of MLKL and a nuclear pore, leading to export of intracellular content into the external space as well as stems. And just want to show you preliminary data showing that the chemotherapy, it can induce a mixture of apoptotic, caspase-1 depending pyroptotic as well as necroptotic cell death. And I just want to leave it as a food for thought, and it's a very exciting time for our group as well as I think many of you, and just want to thank my fabulous group. Without them, I think none of these could be possible, and it's a very exciting journey with them. And also want to thank the funding source as well as the funding source for the trainee. And also at the end, just want to mention that our lab is really recruiting is recruiting postdoc and trainees. So with that, I would like to end and take any questions.
Great, thank you, Keith. That was really a wonderful seminar with some very intriguing data. I'm going to kick off the questions and then go to the Q&A. I will remind everybody in the audience that to submit their questions through the Q&A function on Zoom. So anatomically, Keith, do you know in your xenografts where the PGE2 is produced? Is it produced on the periphery of the tumor to kind of keep the immune response from happening, like a wall maybe, or is it within the tumor itself to make the entire tumor just resistant?
Yeah, that's a good point. So I don't think there is an anatomical per se. So there is a lot of cells that are in the epithelial stromal interaction, as well as maybe in the necrotic region. So so that might fit with the idea that these cells may be, these PGE2 are being recruited, so secreted to recruit other myeloid compartment to play a role. But we need to. But that's a great question. I think my fellow will be better to answer this question. Okay. Yeah.
Good. And along the same lines, then, do you know how long the PGE2 lasts in the tumor? Because the half-life will be, once it gets made, it's not going to keep getting made if the cell is dead, right?
The half-life.
Yeah. How long does it last?
Yeah, we did not look at these are questions we have not looked at the half-life, but you would imagine that one thing I can answer is that we always thought that this is a synchronized type of cell death. Mm-hmm. So to our surprise, the tumors is not as at any time there's a certain percentage, like any time 10 to 20% of proliferating cells. So it's not as we thought in vitro that cell death occur in a concurrent setting. So at any time there will be dying cells, because the proliferating cells are not synchronized. So the drug effects is mainly targeting the proliferating cells. So we think that there's the dying cells also is non-synchronized, happening in a non-synchronized manner. So it's not just the half-life of the PGE2, but also this process that lead to the prolonged effect of PGE2. Yeah,
so it might be a wave as a cell proliferates within the tumor. Okay, so I'm going to go to the Q&As here. We have a question about the cell surface markers that were used to detect the induced tumor cells. Could you speak to that?
Sorry, can you repeat that question?
Yeah, so the question is they hope to learn the cell surface markers that were used to detect the induced tumor stem cells.
I see. So we look at primarily using cytokeratin 14, but we also utilize CD44 as well as CD49F, which are that we didn't have time to get into, but been shown to be stem cell marker by my group and other group. And now more data is coming out suggesting that CD44 positive cancer stem cell might represent a more mesenchymal-like type of stem cell, while the cytokeratin 14 might represent a more epithelial type of stem cell.
Okay, and then another question is there a possibility of pre-treatment necrosis and post-GC treatment-induced necrosis, contributing to chronic neoantigen presentation and eventual exhaustion of the immune cells, including CD8 positive T cells, leading to a poor PFS and OS?
Yeah, that's a very interesting question. So we always thought that, I'm not sure about the audience, but there's a lot of literature out there suggesting that lytic cells that are immunogenic, right? Or necrotic cell that are immunogenic. But there are a lot of controversy. Maybe I take an example using ferroptosis as a type of lytic cell death, that there is controversy that suggesting that while they are beneficial for targeting the epithelial compartment, but they are not, these type of cell death may induce a non-tolerogenic cell death than we thought. So it is, I think it's early days to, I think we need to study the biology of these different type of cell death and their relationship to immunogenicity before we talk about therapy. I think that is a very important point that, at least from my group, that we're working on, and I saw a lot of study out there, and maybe Derek's lab is working on this, can shed some light as well.
So another question we have, Keith, is COX-2 produces other prostaglandins and eicosanoids besides PGE2. Do you think the IDAMP properties are exclusive of PGE2, or are they shared by others such as PGE3?
So I guess, is he asking the specificity of the drug, or they're talking about the biological effect of the other?
I think whether there are other prostaglandins or eicosanoids which are involved.
We have not extend our study into other types of prostaglandins, and we might be extending this to our mass spec analysis kind of gear our study into another direction. So that would be an interesting question, but we have not studied that in our lab.
Okay, good. So then there's a question about the specificity of bladder cancer versus other cancers. The question itself is, does celecoxib have the same IDAMP effect in colon cancer, since it is widely used in colon cancer treatment? And I'll just broaden that question to just other cancers. How transferable is this to other cancers?
Well, as I put a few slides from another group from UK, led by San Diego. So they didn't use the term IDAM, but based on the data of the role of PGE2, COX-2 PGE2 in dendritic cell and T cells, at least they have shown in vivo data in breast tumor. They have other data in melanoma as well as colon. So I would hope that there will be more data coming out to support that this may be a more generalized effect in additional tumor type, as well as a more and to note that they use a different chemotherapeutic drug, 5-FU versus gemcitabine that they're using. So we hope that this is a more generalized phenomenon to additional cancer type as well as additional drug that I allude to at the end.
Okay, then there's a question about your ICD vaccine and whether it induces humoral responses.
Yeah, that's a good point. So our lab is trying to use this multiplex analysis as well as multiplex transcriptomic analysis in both the patient and the preclinical model. So we hope to address that question using a more holistic approach instead of a targeted looking at the certain compartments that we are interested in.
Okay, great. There are two questions here that are sort of related, so I'm gonna summarize both of them. The question is whether tumor associated macrophages may be a source of PGE2, and whether or not the ratios of the DAMPs to the IDAMPs in the tumor microenvironment, have you checked those? And I'm just going to extend it, and I'm just going to ask you, do you know the same cell is secreting DAMPs and IDAMPs, or could it be two different cells with two different responses?
Yeah, that's a great question, right? So this is more a basic science question where you can tissue specific knockout each cellular compartment, right? But in a drug treatment setting, your drug really target the whole microenvironment, right? But long story short is that the PGE2 is known to be also in the, and we have shown that is also could impact the stromal microenvironment, as well as shown by others to be secreted by the fibroblasts, as well as by the myeloid cell compartment. So definitely, but we think that this is a feed-forward effect that arises from the epithelial compartment that feed forward to other tumor microenvironment. So if we can stop this early on, then we can hopefully stop this feed-forward loop that lead to this acceleration of the PGE2 effect in a more global tumor microenvironment. I don't know. I hope I answered that question.
Okay, I think you did. Another question is, could high COX-2 cancer cells, I assume expressing cancer cells, represent more resistant cells that get selected under therapeutic pressure? So you're selecting for cells that have high COX-2 expression over time.
Yes, that's a good question. So at least from the pre-treatment patient tissue that we mine in the ICI treatment in MVICTOR-210, I believe, pre-treatment, so a high expression of COX-2 or PGE2 in the tissue has a predict response to ICI, as well as so yeah, so that's the data answering your question, I believe. But obviously we need multiple cohort and additional patient tissue to hopefully also sequential tissue from certain treatment to address your question. But
Dr. Abbott, that's probably a good place to end our seminar series today, our webinar series presentation. So again, thank you, Dr. Abbott, for excellent moderation. Thank you, Dr. Sisson-Chan, for an excellent talk. I'd like to remind everyone next week we have a dual sponsored seminar from the Cancer Moonshot and the Seek Space webinar series. Camien partnering with patients to the land to Camien partnering with patients to define the landscape of rare aggressive sarcomas by Dr. Janeway from Dana-Farber Cancer Institute. So again, thank you for an excellent webinar today.
all right.